So what I'm going to do is basically present to you guys how to use biological databases and some of the tools that are available for you. And this is more of a beginner lesson because we only have a short amount of time. And this should at least give you an idea of how to use some of these databases and a little idea of what sort of tools are available on them, and really introduce you to the massive amount of databases and tools that are available to you. So I'm part of the measurement committee. My name's Ian Schlander. I work for the National Renewable Energy Lab. And I work as a metabolic engineer there. And I was an iGEM team leader. I was an iGEM judge last year. And now I'm part of the measurements committee. And so here to help you guys. So the databases that we're going to cover today will be BioCyc, which basically sequences has sequenced genomes with predicted metabolic pathways. We're going to go over Brenda, which is the enzyme database. We're going to look at UniProt, which is the protein sequences and functional information of these proteins, huge database for proteins. We're going to go over PDB, which is a protein database or protein structural database. And this gives you all the structural data you could possibly need for a protein that is known. And we're going to go into NCBI, which is mostly for biotechnology. And we're also going to go over GenBank a little bit, which goes a little bit more into DNA sequences and nucleotides. Some tools I'm going to demonstrate today as well will be NCBI, the BLAST, which is basic local alignment sequencing technique. And that aligns homologous sequences for finding a protein of interest. We're going to go over the GenBank RefSeq, which shows you how to find a DNA sequence of a protein in the genome of an organism. And then we're also going to go over some example tools that are available, like Clustal Omega, which aligns multiple sequences that you get from your BLAST. And you construct phylogenetic trees to show the relationship between these and how related they are. And then we're going to go over XPASI, which is almost a database for bioinformatic tools and resources available for you. And so by the end, hopefully what we'll be able to do is, you'll know what databases are available, how to navigate the database. That's our number one goal. And then number two will be to use the database information to decide on sequences of proteins from different organisms and how that's really useful for your project. And then three, using bioinformatic tools in conjunction with the database. So you can analyze and visualize these proteins, for instance, and basically look at pathways. And really what the goal of my bioinformatic tools is to use the analysis that you get from these databases and decide on a protein that really you want to use. And so this is the flow chart that I'm going to reference throughout the talk. But what we're going to do now is go to I'm just going to demonstrate these tools. So we're going to start with BioCyc. So what BioCyc is basically, it's a collection of 16,822 pathway/genome databases called PGDBs. And what I really want you to know is these 3 points right here and how it's useful for you. And so what you can use is it's quality data curated from tens of thousands of publications. It's computationally predicted metabolic pathways and operons. So from this data that they get from researchers' posts, they will start to predict metabolic pathways using the software called Pathway Tools in BioCyc. And then they will integrate data from other databases. That includes things such as how is the gene essential, the regulatory networks of that gene, the protein features, and Go annotations. And so one thing that I do want to highlight real quick is BioCyc is sort of the main site, while it does hold several different sites within inside it. And so for instance, BioCyc is exactly what I just explained. It's a database collection, but then you have something sort of cool, which is something like EcoCyc, which is just an E. coli database. So it's for model organism, E. coli, and you can basically look at all of the genes, all the known things about E. coli, and also predicted metabolic pathways. You can also look at MetaCyc, which is a really cool database that goes into, which is the important part, if you want to look at, is basically it's an encyclopedia of metabolism. It predicts metabolic pathways and sequenced genomes. So again, they're sequencing the genomes and they're predicting the metabolic pathways based on these sequenced genomes. And then it's really to support metabolic engineering. Let's say you want to take an enzyme and you want to know what it does, what metabolic pathway it goes. You want to put it into another organism. You really want to use MetaCyc for that. And really, it's BioCyc is invaluable tool I use in my work all the time, and I'm so thankful for it. And so really you can use things such as a genome browser. You can look at the metabolism of the cell, but we're going to save that for later. And what we're really going to go into is EcoCyc, because we are iGEM and most of the teams do use E. coli. And what we're going to do, for instance, is one thing that's really cool is you can either look up, enter a gene, a protein, a metabolite, or a pathway. And so what we're going to look up is, let's say we want a monooxygenase enzyme, something that has a classification of enzyme for monooxygenase. And so what it's going to give you is different pathways involved with that. So 2-component alkene sulfonate monooxygenase. And then it's going to give you proteins. It's going to give you gene ontology terms. And what we're going to do is we're going to click the first protein that is known about that. And so what it's going to take you to is basically the original summary page. And what you can see here is kind of the transcriptional unit here of the regulation of this gene. It's called UBH, highlighted there. And what it's going to do is to give you a summary and basically tell you what this gene is known about in a real short and sweet way of understanding what this gene does. And it also gives you things such as Go terms, which can tell you the biological processes, and it'll take you to different databases and what is known about that. Tell the essentiality of the gene, basically, and how what knockouts affect it. It can also tell you the reaction. So this is an enzyme, for example, that has an enzyme on this sort of molecule. And it'll show you the reaction right here. It'll also tell you the pathways that is involved that you can click to, and it'll take you to those pathways. You can also see the protein features. So mostly you can see what the exact amino acids that are part of this protein. It'll show you the operon. So you see a UBH here. You can zoom in, zoom out. And so it'll show you the terminator involved. It'll show you the promoter involved and all the proteins around it out of its native orientation in the genome. And it'll also show you the references in case you want to look at the literature further and look at what is known about this gene exactly. So it's an extremely useful tool when you are looking for something like a monooxygenase. You want a specific reaction. You can look up this reaction and it'll bring you to different types of enzymes that can do that reaction. So what we're going to do now is we're going to look at UniProt. And so UniProt is a huge protein database. And what we're going to do is we're going to look at the EcoCyc, the EcoCyc accession number. So there's a list of accession IDs right here. And what you see is UniProt. And so what we're going to do is we're going to copy this number. And we're going to paste it into UniProt, and it's going to bring us straight to the exact protein we were just looking at. And this gives you, again, more information rather than just exactly the metabolic network and everything, but it tells you exactly what the cofactors are for this protein. It gives you the molecular function, exactly what it does when it binds to different binds to other proteins and cofactors. It's going to give you the biological processes that it's useful for in case you want to use synthetically produce ubiquinone. You can definitely use this enzyme. It has tons of information, the names, the taxonomy, to go further into exactly where it got it from. So it gives you the location where it localizes to, the pathology of it. Again, this is just an extremely large database on proteins. So this is just an example of one protein, and they have tons. So that's an example of UniProt. And so this is an enzyme. And so what we're going to do is we're going to type in VisB, because that's the name of the protein we were just looking at. And so it's going to bring me to all the search queues. And so give me only one for this protein. And so what you see for this enzyme is it's going to show you the reaction scheme of exactly what it does, exactly what you need inside the reaction, and what to expect from the output of the reaction that this enzyme does. It's going to tell you taxonomic range, which is E. coli. So if you are working with E. coli, you would want to put this into your chassis E. coli to use it. It'll also go more into the reaction. It'll give you a reaction diagram here as well. And it'll give you the pathways that it's involved in known to the database, which is right now ubiquinone biosynthesis. And then let's say we want to look at PDB. So this is the protein data bank, and it gives you more of a structural view of exactly what the proteins are that we're looking at. So for example, we're going to look at UBH, which this is the gene for the protein that we're looking at. And so there's two different genes. And so what you can see is the organism that it comes from. So it comes from, this one comes from Pectobacterium atrosepticum. It's a mouthful. And this one comes from E. coli. So this is the one that we wanted. We're going to look at this. And so this is a really useful tool in case you're looking at what exactly part of the structure, and let's say you're enzyme engineering or you're engineering a protein and you want to only look at what parts are interesting to you. It'll give you the literature that's involved with this. It'll also give you the macromolecules that are also involved with this. The small molecules, the experimental data and the visualization of that data. Another cool thing that it can do is you can look at the 3D view of this protein. So it gives you the amino acids here. You can select the amino acid here. Let's say we want to look at Q, the second amino acid, and it'll start to highlight that amino acid right there. And so you can really look at the structural data of a enzyme or a protein that you're engineering, or you're just interested in knowing what exactly it does inside the organism that you're engineering when you put it inside there. Let's go back. You can also look at the annotations, which will tell you about the different types of architecture of this protein, the domains of it. And you can also look at the sequence, and it'll explain to you exactly which part of the sequence and what structures it has. For example, this one, if you look at the key at the bottom, let's look at the yellow arrow, which will be a beta strand. It starts off with a beta strand. So you can look at you can really get into the structure of a protein using PDB. The only thing about PDB is it has to be a known structure. Therefore, if you are using EcoCyc to find a new protein on a, let's say, a metabolic pathway that is computationally derived, but there is no structural data on it, then you will not be able to use PDB. So here's what I'm going to show you what you do when, let's say, you still want to look at the structure further and it's not on PDB. So what we're going to do is we're going to go to NCBI, the National Center of Biotechnology Information, and we're going to look up VisB, and what it will do is going to give us all the VisB genes and protein, or VisB proteins throughout different organisms. But what we really want to look at is still stick to E. coli. So we're going to stick to E. coli K12. And so what it's going to give you is a bunch of literature, the genes, all the stuff on it that is found in the database. And what we're going to look at is the protein itself, and it's going to give you a list of 23 proteins. I'm going to click on the first one, the protein we were looking at earlier, and basically it gives you sort of the data on the protein inside the database. So what we're going to do is we're going to run a blast. And so see what it says in the caption here, it's going to find regions of similarity between this sequence and other sequences using BLAST. Let's say you want to find a protein or an enzyme that does a similar reaction, but you don't exactly want this one, or you want to see if it's in another organism that you're interested in. So it's going to take us to this page when we click BLAST. And so under organism, all you're going to do is just type in eubacteria, because you want to keep it inside the eubacteria taxid. And then what we're going to do is we're going to exclude E. coli because we want to look at other organisms that might have this protein and not E. coli. So we click the exclude, and we click BLAST. It will take a few minutes, maybe 5 to 10 minutes, sometimes maybe a minute if you're lucky. But I already did the BLAST before this, so this is the page it'll bring you to. And so basically what you're looking at is the max score, and it's looking at just the homology between the protein VisB without showing you the E. coli. And so we're going to see is percent identity. So this one has 100%, so that's really good. That means that it probably has the exact same protein inside a different organism, in case you wanted to use a different organism for a reason. So what we're going to do is select the top 3 that have the closest homology. We're going to click download. We're going to click complete sequences. It's going to give us a seq dump file. And so what this is this is the FASTA format for the protein. So these are all the amino acids for the third one, all the amino acids for the second organism that we saw, and then all the amino acids for the first organism we saw. And so, for instance, what we're going to do is now we want to really see the similarities. We want to align these sequences. So what I'm going to do is I'm going to copy all of these in FASTA format. All these, most bioinformatic software will ask you to put the amino acid sequence, the protein sequence in FASTA format. So in case it's ever a problem, definitely look that up. And what we're going to do is we're going to go to Cluster Omega. And so it's a multiple sequence alignment tool. So you can align your sequences and you can see what the differences are between each one. And so then you're just going to click submit. And it's going to take you to this page. And so what it's doing right now is you have your first, second, and third organism, and it's going to show you the alignment between each one. And what I like to do is I like to show colors. So you can see all the little differences between this one. See, for example, the first, or on this exact amino acid, this is where the difference is between the first organism and the other two. You can see between the gap in the stars as well here. And you can see that these sequences are extremely well aligned. Usually when I do a sequence alignment, you're going to see a lot of differences between different organisms on different enzymes. So this is a really good sign if you want to take this enzyme and put it into a different organism. But again, let's say that one of these proteins do not have a are not known inside the protein database, where you can't understand the structure, you don't really know what's going on with it, and you really want to understand what's going on. So what I'll take you to is XPASI, and it's a bioinformatics resource portal. And so what I'm going to click on is find resources. You can also queue all databases, which will take you to, which will look at all these different types of databases. But what we're going to want to look for is a resource that can help us analyze this secondary structure. So I'm going to type in secondary structure prediction. And so it's going to give me a bunch of different resources that have been software that have been created, and a bunch of tools that are available for biologists to basically analyze secondary structure and look up and basically predict it. And so one that I'm familiar with is CFSSP. And so this is a secondary structure prediction server. And what you're going to basically do is you're going to enter a protein sequence. So we're going to go back to this format. Let's say we want to analyze this third one. You're going to want to highlight the entire FASTA format. And then let's say predict. And what it's going to do is it's going to basically show us all the different structures and part of this protein that it predicts. Now, I will say that I have found several times this is a varying software in its accuracy. It cannot predict tertiary structure as well. And this is just to give you a better idea of what you're looking at of the protein, if in case you do not know what it is at all. So that's where it becomes important that you use multiple sequence alignments so that you can compare. Let's say you have an active site at residue 378. Let's say you have an active site there that's really important to your enzyme. Well, when you put the excuse me, this, I think I'm getting the same thing you're getting, Alexi. It's not letting me move this. Ah, there we go. Okay. So let's say you go back to the sequence alignment for Cluster Omega, and let's say this is an important active site right here. You will definitely want to look at the secondary structure. You want to get as much information as you possibly can if it's not available in PDB. And this will allow you to sort of make inferences on the differences of the active sites. It'll make you inferences on what the differences are between the proteins and really make a selection on which one sounds best for your project. And let's go back to the NCBI. Let's say you do choose your protein, and finally you want to, start messing around with it in SnapGene or whatever software that you use to engineer your plasmid and basically begin the lab to enter the lab with it. So what we're going to do is type in FISB E. coli. And so the first thing that pops up is the gene UBH, which is the gene for your FISB. And what you're going to see is a summary of the gene, and the more important part is sort of the genomic context. So it tells you exactly which part of the genome it is by these little nomenclature right here. So out of 3,000, 305,000, or 3,055,000, it's between these two parts inside the genome. And so what you're looking at here now is UBH right here, RefSeq. And so what you can do is you can zoom out of the genome, and you can see it more in context of other proteins. You can zoom in case you want to look at more closely to the, let's say you just want a certain sequence of this protein. But what we're going to do is we're going to highlight this region, and it's going to bring up a little screen right here. And what we can do is download the FASTA. So this is going to bring us a FASTA file. Excuse me. Let's try that again. So we're going to download the FASTA. Just trying to bring it up on my structural software. But basically, this is what it's going to show you. I pulled it up on my Microsoft Word document, and this is the sequence that I highlighted. And let's say now I can copy that sequence and I can put it into SnapGene, and I can start putting promoters behind it. I can start putting terminators behind it. I can start constructing my genetic circuit. And so basically that's why RefSeq, I use it all the time, and just put it, it's really simple. SnapGene opens it up right away, and you can immediately paste it into your SnapGene. And so basically what we did, if we want to recap what we just went through, is we looked at BioCyc, and we saw that there was different, we saw the EcoCyc, we looked at Metabase or MetaCyc. There's also HumanCyc that looks at metabolic pathways of humans genome. And we chose the monooxygenase. We just wanted a monooxygenase started from the beginning. We then analyzed monooxygenases using UniProt. We looked at the VisB in UniProt. We looked at VisB in Brenda, and we looked at the structural data of VisB in the protein database. So then we wanted to then start messing around with the sequence and start analyzing more. So we went to NCBI, and we looked at the we took out the exact sequence from GenBank using the RefSeq tool that you can now put into your SnapGene in case that's the enzyme that you do choose. But then tracking back, we first went to NCBI BLAST, and that's where we align the different enzymes, so that way we can look at all the different enzymes that we wanted. And then we use Cluster Omega to then then analyze the sequence alignments and see, let's say, the different active sites of the enzyme VisB. And then we use XPASI bioinformatics tools to then look at the secondary structure of a proposed protein that we didn't know what the actual structure was. And again, I do want to say that this is a variable software. When you're looking at prediction software, it is not always accurate, so do not take it as the holy grail. It is something that you have to take with a grain of salt. It is just guiding you towards what you could be looking at. So that's sort of the main workflow when you're using a database. You can look at genes, you can look at specific reactions, you can look at, if you want a specific metabolite, you can look at all the different types of enzymes, the metabolic pathways inside there. And then you use these databases to basically analyze it further, make decisions on, is this the exact enzyme that I want, and start to narrow down your search. And then you use really the BLAST tool to sort of compare the different enzymes of your list of the exact reaction that you want. And you use these tools to sort of further analyze whether sort of single down to that specific enzyme that you want. And then you transfer over to GenBank and RefSeq to actually grab the sequence out of the database and start to put it into your design software. I hope you guys enjoyed that. Now what I want to shift to is time for you to practice. So what I want to direct your attention to is the database and bioinformatic tools for beginners practicum. So what I did is I basically developed a little exercise for you guys to practice these sort of tools and softwares that I've explained inside this webinar. And you can find this, the download at the download document on the webpage for iGEM measurement webinars. You can copy and paste this into your browser, and it'll take you straight to the webinars that the measurement committee is doing. And if you look under this specific webinar, you'll see a downloadable file for this exercise that I want you guys to practice and get better with these tools. And so if you have any questions after you do this exercise, or if you have any questions about this specific webinar, please contact measurement@igem.org and request Ian Schlander, that is me, for further questions. I'll be here to answer any of your guys' questions, and I would love to help you guys out with any of them. And what I do want to do now is allow Sonia Billerbeck to discuss cloning strategies. Before she starts her webinar, I do want to look at the chat and see if anybody is asking any questions.
So there's been a question for you in about what the score is when you do a search in the different databases. I don't remember on which one the question was asked. So where would you see the score of your search?
The score of my search.
When you use
on BLAST. In BLAST.
Yeah. The BLAST. Okay, apologize. That's a good question. So right here, right after I click BLAST, right? So I did the BLAST, click BLAST, and you want to do a protein-to-protein BLAST. Sorry if I did not mention that. Thank you for having me review it. And basically, it's going to bring up all of these different alignments, and it does a good job of immediately aligning from first to last the most closest homology to the sequence that you have of interest. And what you're going to see is a query cover, which will show you all the different. Really, what you want to pay attention to is the query cover and the percent identity. These two numbers are the numbers that you want to have closest to 100. That way, it means that this is the exact protein inside a different organism, and that way you can actually put this inside your a different chassis if you wanted to. Let's say you didn't want to do E. coli. So these are the percent identity, and that's really the number that you really want to pay attention to. And yeah, so once you do that, you can basically start moving over to RefSeq, or you can analyze it into Clustal Omega. I'm going to stop sharing my screen.
And we had another question from Sharon, which says, when is non-redundant protein BLAST used?
So you'll want to use a non-redundant protein BLAST when you don't want to include the exact same organism, or when you don't want to include the exact same sequence that is aligned inside your BLAST. And for example, let's say you have this VisB gene inside your E. coli, but with your circuit, it can't metabolically use the enzyme. It creates a metabolic burden. So what you're going to want to do is basically use another protein that is not as close to that protein, because clearly it causes a metabolic burden, and then use the non-redundant protein BLAST, and it'll start to bring up cues of different proteins that are closely aligned homology, but not exactly 100% identity. That way, you know you're getting a different protein with possibly different effects on the cell, but you know it's also very similar to the exact reaction that you want it to do. And that's when you would really try to use a non-redundant protein BLAST. Hope I answered your question effectively. There's another question that says, could you explain what a Go feature is a little bit more? Yes, no problem. So I'm gonna share my screen for that real quick. Okay, so Cole Dwiggins, could you explain what a Go feature is a little bit more? So for example, if you go to E. coli, and let's say, or we were over here. So the Go terms really is simply just taking you to the gene ontology. So it's really telling you kind of where it's from, what it's used for, the synthesis. It's really giving you the classification further on what the enzyme is. So for example, we use the Go term for biological process. Let's say you want to use ubiquinone's biosynthetic process. So these are all the databases it's gathering its information from. And it just gives you the gene ontology. So it just gives you basically the classes, the different team members. It would basically give you the families of the protein in case you wanted to know further about the protein, but you wanted to look at its the different classes that are related to this protein. You could definitely use the Go terms. That's how I would use it. Again, child classes, like let's say it's a different, this is a different reaction that this enzyme does. This one is a little bit of a different metabolic process and the parent classes. So let's say we click on the parent class, and it'll basically start to tell you which, oh, it's not going. But yeah, it's basically just the classification. So it would be useful if you wanted to use an enzyme that uses a similar reaction, but not the exact same reaction. You want to look at similar enzymes of a metabolic pathway. That's when you would really be interested in the Go terms.